Hello and welcome to BIOS Frontier Science. Today we're thrilled to welcome Dino Di Carlo, Professor of Bioengineering at UCLA. Dino, thank you once again for joining us. To kick things off, could you please give us a quick introduction to yourself?
Yeah, sure. Thanks, Gage, for having me. It's a pleasure to be here virtually. So I'm, as you mentioned, I'm a professor and vice chair of the bioengineering department at UCLA. I've been there since 2008. And my background in my lab works kind of at this interface between micro and nanotechnologies and biology, and how do we use these micro and nanotechnologies to automate life science research, develop better diagnostics, develop medical devices. So I've throughout my career, I started off as a bioengineer, actually as an undergraduate at Berkeley. Continued on through my PhD in bioengineering at the Berkeley UCSF program, and then spent a few years actually at Mass General Hospital in a clinical environment, but also in an engineering lab in that clinical environment. And that, I think, really had a big impact also on the thinking that we have in terms of, how do we make simple things that can have translational impact, that can make it into the clinic, help patients.
Awesome. And before we dive into some of the projects your lab is working on, your website talks about reaching the ultimate limits of biology. Could you tell us a little more about this and what it means to you?
Yeah. So the way I think about that is, in physics, right, you have the quanta of physics, which are atoms, but you can break those down into quarks and things. But there at some point there's a quanta there that's the interesting thing, and you can't break it down further. In biology, that quanta is a little bit different. The quantum kind of metric there is in biology single cells and single molecules, right? So if you think about it, we want to understand biology at that level. If we want to measure what a single cell is doing, we don't cut a cell in half. That's not very useful or interesting. And we want to measure, a single DNA molecule or a single protein molecule. Cutting that in half or breaking that apart further is not as interesting anymore. So that's how when I discuss really the ultimate limits of biology, I'm discussing those key quanta that we want to study, that we want to measure, and that really, we have the tools now to do, and you don't want to go further. So that's kind of an exciting aspect of where we are in our understanding of our tools to understand biology, is that we're really at the limits of what you want to measure.
Yeah, and so your lab has put a lot of work into developing these tools. And so one of the first things we want to talk about is inertial microfluidics. So this is a term your lab coined, but before we dive into the specific projects you're working on, could you tell us a little bit more about this term and what inertial flow is?
Yeah, it's a great question, and partly it's because this term came up because the microfluidics field, and the field that I grew up and was studying in college in microfluidics, we always heard that that flow behaves in the way that is like honey, right? That there's no really inertial effects, where things like turbulence or things like recirculations that you might expect if you look at a river, or if you look at these large scale flows. And that's because this idea, there's this concept of a Reynolds number or kind of this ratio of how important momentum of the fluid is versus the viscosity, that was always low in microfluidic systems. And you shouldn't really rely on using inertia for anything. And so that's the paradigm that I grew up in. And even I remember on qualifying exam that I had questions about this, and I would say, of course, there's no inertia. It's just all with Stokes flow. That's what we refer to it as. And that was the right answer at that time. And so what I questioned was that we couldn't use inertia for something useful in microfluidics, and it turned out that we could. It started off with some accidental discoveries of seeing how particles behave in these microchannels in ways that was unexpected. But in the end, what it ends up being is you can use the momentum of fluids to do really cool and important practical things in microfluidic devices. for example, position cells and flows to either analyze or separate them, or separate out different populations of cells, or even shape the flow itself to create particles and other structures. So I think that the concept of is a long-winded response here, but the concept of inertial microfluidics is really that we can use momentum of the fluid, even in these small scale systems, to do really practical and useful things.
Awesome. And so you mentioned a couple of use cases around cells, but could you dive a little deeper into how this property of fluids could be used in microfluidic devices?
Yeah, so one of the key ways that we use fluid momentum is in positioning cells in microfluidic channels. So, one of the kind of ubiquitous instruments in life science research is the flow cytometer. This is, ways that it's used in clinical diagnostics. It's also used in life science research, where you measure one cell at a time as it passes through a laser. You characterize some fluorescence or other properties of the cells. What that relies on is first that you align cells so that they kind of go single file through that laser spot. And so what we found is that we could use fluid inertia to do those alignment steps, to have cells that started off randomly distributed in a channel kind of self-organize and align into a narrow stream. And that was super unexpected, right? So you can imagine if you put cells through a channel and they're randomly distributed, you might expect they come out randomly distributed. But what we found and really pushed forward and made practical is that you could, by changing the kind of the shape of the channel, that you would see that those randomly distributed cells would align to subsets of locations in the channel. And if you change the shape in a very particular way, you could get it down to single positions in those channels. And then you could start to analyze those cells very, very nicely. They're all in the same focal spot, always being excited in the same way, all moving at the same velocity. Beyond just focusing behavior, what we've also found was that cells kind of moving down these channels would also order. So there would be a spacing that was preferred. So instead of like randomly distributed as they go through these channels, they would also organize into organized like trains. And that also is really beneficial if you think about flow cytometry. You don't want two cells to be in that spot at once. And so this is a way to kind of structure the flow, and kind of space out cells so that you can measure more per unit time.
Interesting. And so what is the advantage of building devices that use this property, versus devices that use other mechanisms for sorting and organizing cells?
Yeah, so the key advantage is that it's very simple, right? So you don't have other fluids. So in the case of flow cytometry, you often have like sheath fluid, or you have some other force like electric fields or magnetic fields that you have to impart in the system in order to organize or separate out cells. And so this was an intrinsic process. These inertial effects happen just by flowing cells through a channel. So it's in some ways, it's this elegantly simple approach to solve this problem where you're not like over engineering the problem, right? You just flow cells through a channel of a certain shape, and they behave in the ways that you would require all these external instruments, either additional sheath fluids with pumps, or you need magnetic fields or other fields to do separations. And so that's kind of a key advantage. And that also allows for scalability. And what we're seeing now is that several companies that are building off of this inertial microfluidic technology to do parallel flow cytometry, right? Because if you don't need all these external things to control your flows, then you can parallelize it much more easily. And there are also parallel separation to do separation of white blood cells from background, or even specific subpopulations of white blood cells at really high throughputs. And so there's companies that are developing technologies for cell therapies, for example, to prepare particular subpopulations of lymphocytes for downstream use.
And so you actually helped found a company in this space called Vortex Biosciences. Could you tell us a little more about what they're working on specifically?
Yeah, so Vortex Biosciences is again using some of these inertial effects. In their case, they use what we call recirculating flow. So if you flow in a channel that expands suddenly, the flow doesn't follow the expansion, and so you get this recirculating region. And what we found in my lab is that larger cells, like cancer cells, would accumulate and capture in these recirculations, while smaller cells, other blood cells, would flow through. And so what Vortex Biosciences is commercializing is a cell, or has commercialized and selling product now, is a approach to isolate larger cells from a large background and do that really high throughputs. Again, this requires flowing fast, so which is a good thing when you want to do cell separation from a large volume of blood, let's say. And so one of the key areas where this is, where Vortex Biosciences is working, is in isolating circulating tumor cells. So these are cancer cells that have sloughed off of it or escaped into the bloodstream from a primary site or other metastatic sites, and they're circulating. And so what we found is that in many patients, in particular epithelial tumors, that you get large circulating cancer cells that you can isolate in these vortices, and then you can release those very easily into small volumes to perform analyses. In particular, one of the key things that is of big interest is being able to understand what mutations are present in those cancer cells. What particular mRNA, do they have a splice variant of, androgen receptor, for example, in prostate cancer patients, which suggests particular types of treatments. The androgen receptor signaling inhibitor kind of treatments would be more efficacious in those patients.
So it sounds like cell sorting and cell separation are some of the early applications for this technology. But where do you see these types of devices in 10 plus years? What will we be using them for?
Yeah, I mean, I think the way to think about that is in cell separation space and cell analysis space, there's always a need to manipulate and kind of position cells for downstream either imaging or some downstream type of analysis. And so I guess what I see is that, and we're seeing this already with some of the companies that are building off of the technology, is that this is a very simple way to do that without a complex system. And so that allows for kind of higher throughput types of operations. And so I guess what I probably envision is going to happen is, as you look at companies that are doing image-based cell analysis now, and you're seeing more and more of these companies are image-based cell sorting, whether it's for diagnostics, whether it's for life science research. All of these types of companies will probably incorporate some of these inertial microfluidic approaches, because it's just easier to get what needs to get done, which is position cells and manipulate them at really high rates.
Awesome, that sounds fantastic. So moving on to another topic that your lab is working on, smart biomaterials. Could you give us an overview of what projects you have ongoing in this space?
Yeah. So this kind of topic area arose from our expertise and interest in microfluidics, and using microfluidics to make biomaterial-based particles. And so microparticles that are hundreds of microns or less in size. Really at the size scale of cells. And so, kind of traditional biomaterials, you think about hydrogels that you might inject, that there's a fundamental trade-off in those materials, and that if you can inject them and therefore they're easy to use in vivo and for treating kind of tissue wounds or defects, they become kind of a barrier for cells to infiltrate and integrate, right? And so you have this kind of trade-off where you have materials that are just filling, are kind of fillers, but not really integrating with the biology, or you have to add some type of growth factors or some other kind of exogenous factor to help cells kind of integrate and interface with the material. And so what we found is that by not including growth factors, but just by structuring the material in a particular way. For example, our materials are mostly these slurries of these spherical particles. So you can imagine it as like a gumball pack of spherical particles that you can then flow, but then they also can set and interact with each other and the surrounding tissue. And when they set, now you have all this porosity. You have all these cell scale pores, so that cells can infiltrate and get into the material. And that really creates this interface with the body that's much more seamless. And the cells that infiltrate first, which are usually immune cells, they secrete all of the growth factors and signals you need for tissue to regrow, for other cells to get in that are creating blood vessels and fibroblasts to get in, et cetera. So it's really smart in that we use the body itself to create all the signals it needs in order to recreate the tissue, rather than try to force it by adding our own growth factors, which we never can really do at the right times, right? Because this is also a dynamic process during healing.
Interesting. So you developed a material that remains porous and is still easy to use, so it allows cells to infiltrate. But what do you hope that will kind of enable? What applications do you think that this type of material will be used for that existing hydrogels can't?
Right. Yeah, so what we've demonstrated in the lab so far is applying this type of material in kind of healing, whether it's epidermal wounds. So these are big defects, right? So you can, in diabetic patients, for example, they have these large defects that don't heal well. After surgeries, when you have large pieces of tissue that are removed, these are things that don't heal well. And so filling that tissue and allowing new tissue to regrow in becomes a really important application where there's not a good solution right now. Beyond that, and I mentioned that these materials also instruct immune cells to enter and interact with them. A recent paper, we demonstrated a lot about how we could actually tune the types of immune cells that entered. And if we got a more of an adaptive immune response, where you have immune cells that are actually recognizing parts of the material, that enhanced healing even further. But that also led us down another path, which was, okay, if you can tune the adaptive immune system, can you use this in sort of vaccine applications where you want to deliver antigens over prolonged periods of time in order to really tune the adaptive, the types of antibodies that are generated, the types of T cells that are being generated from towards the antigen that you're releasing. So we have kind of ongoing projects also in the vaccine area using these smart materials.
Interesting. And we'll get to some of the future directions in a second. But before we do, you helped found Tempo Therapeutics in this space. Could you tell us what they're working on and kind of where they are right now?
Yeah. So Tempo Therapeutics is developing and commercializing what we call MAP technology. It's microporous annealed particle technology. So again, these flowable spherical hydrogel particles that then can kind of get flown into a wound or a surgical site, annealed together so that they all kind of form this structure that's stable and allows cells to infiltrate. So Tempo has done a lot of work on manufacturing these, has done a lot of studies in canines and actually some felines as well through kind of vet practices. Because this material, there's no growth factors or things, it's actually a medical device. The FDA has indicated it will be classified as a medical device. And so they were able to work really closely with these real world cases of animals that normally are getting like limbs amputated, right? Because they say they have a tumor, that tumor is resected, but the wound is so large that there's no way to really let it heal. And so they put their material over the wounds, and they're getting really great rates of all the animals that were treated. They were able to regrow skin, regrow hair over these large like resections of tumors. And all this while they're still walking and being at home, right? So this is kind of a real world environment. Now they're working through the kind of the final processes to submit to the FDA to get clearance to use this in humans. They will also probably through So they're also working on in-human studies through a investigational device approach as well in parallel. So that's kind of where they're at. It should be in humans soon. We're hopeful. It seems to be an amazing material.
That is amazing. And so going back to a few of the things that you were mentioning right before Tempo, what future directions does your lab hope to explore in the space? You mentioned a few, but would love to hear more details about them.
Yeah, so I think I mentioned the vaccine area. Because there's this ability to modulate the types of immune cells that get into the material, and you have this huge surface area to present antigen, this is a great depot to deliver antigens for recombinant antigens to generate very precise immune responses. We're also able to, what we found is we're able to tune the type of immune response. So in some cases you want different classes of IgG, for example, that can be neutralizing or are better for antibody-dependent cell killing. And so what we found is by tuning the materials and the types of things that were in the materials, we could get them, we could get more titers of IgG1 versus an IgG2 isotype. And so these are kind of proof of concepts that I think are very exciting. And so that's a direction that we're moving in. I think there's a lot of other kind of tissue generation applications for the material. So, if you're trying to bulk up and create new tissue, this is a great way to do it because you have, you create the scaffold, but you also create the ability for the cells from the patient to grow into the scaffold from day one, right? There's no degradation required. And so that allows you to grow new tissue. And so we're applying that beyond kind of skin wounds into a number of other tissue types. This is, also, it was a great collaboration in my lab from another postdoc, Don Griffin, who's now a faculty member at University of Virginia. He's continuing on in many other directions with the technology there as well.
That's amazing. Excited to see where it ends up. So moving on to another project or another topic that your lab is working on, you guys are developing tools to measure the mechanical properties of cells and using it as a method to diagnose diseases and discover new therapies. Starting with diagnosing, could you tell us a little bit more about this effort?
Yeah. So, normally when we think about analyzing cells to diagnose disease, I go back to the flow cytometry example where we're measuring some surface marker on maybe leukemia cells that tells us, okay, these are leukemic cells because they have this protein on their surface. What we haven't done, and there are no diagnostics like this yet, is let's measure, what's inside the cell. What is its structure and how does that change and how can that be used diagnostically? And that's a bit harder to do, right? Because you have to, it's kind of an integrative measurement of many things, but it can be really important. So you can think about examples when, immune cells become activated and they have to migrate into tissue, they're changing their mechanical properties. Or cancer cells that are very invasive and are metastasizing, they have to have also different mechanical properties. And so you can imagine that parameter, this kind of holistic parameter of how stiff or how squishy a cell is, that could have diagnostic value. And so that was the direction. We started this project maybe over 10 years ago now on kind of how do we, it was known, that mechanics of cells was important from a biophysical perspective and it connected to disease. But really being able to connect that to a practical system to measure it for diagnostics, that was kind of out of the question at the time. So that's kind of the direction that we went. And what we did is, again, we took advantage of inertial microfluidics that allowed us to align cells so that we could have them get squeezed basically by another flow of fluid. And then we used high-speed microscopy to analyze as those cells are being squeezed. And we used machine learning to analyze those images. And that allowed us to measure thousands of cells and their mechanical properties. And then from that, now we were able to diagnose disease states. And so that's a project that's that's kind of spun out to commercial efforts now as well.
Interesting. And so what advantages do you diagnostics based on mechanical properties have over some of the more traditional methods?
Yeah, so the key advantage is speed. You can just make these measurements extremely quickly. There's no, if you think about it, if you're going to measure, biomolecular properties of cells, you either have these labeling steps with antibodies, washing steps, you have all this sample preparation, you have all these incubation times. And so if you want to make a measurement, in 5 minutes, you can't do that with those technologies. Or if you want to look at, RNA, or you want to look at DNA, you have to lyse cells, you have to amplify, all those to also take a long period of time, right? Even the kind of the most rapid, analysis systems that use like isothermal amplification for, SARS-CoV-2, they're like 20 minutes or so. But nothing is like in 5 minutes or less, right? So that's where these mechanical measurements can be extremely rapid, because there's nothing you do to the cell, right? You're measuring it in its kind of unadulterated form, and you look at what's happening, and you can make an analysis and report that back. And so that's most important when you have emergency conditions, infectious diseases, and things like that. And that's where it's being applied now. By Avail, which is the company that's commercializing the technology.
Interesting. And so you're also looking at mechanical forces as a discovery tool for kind of discovering new therapeutics. Could you tell us more about how you're going about this?
Yeah. So you kind of beyond kind of the structural aspects of cells also apply forces to their surroundings, to other cells. This is more ubiquitous than you might imagine, right? Obviously, we all know like our skeletal muscles, cardiac muscles, and other smooth muscles, but every cell in our body applies force. Immune cells, when they interact with another cell that is presenting antigen, they have to pull and tug on it in order for it to effectively activate the immune cell. So there's forces everywhere. So the challenge, and there are techniques that were developed in the academic community to measure forces that cells apply, but one cell at a time kind of ways, or maybe a few cells. And so what we worked on is like, how do we know that this is important in biology. We know that there are diseases that are related to improper amount of forces that are generated. I can give you many examples, whether from like things like fibrosis, where you have myofibroblasts that are squeezing too much and stiffening tissue, to incontinence, where you have the bladder smooth muscle cells that are not squeezing the correct amount, to preterm labor, when you have the uterine smooth muscle that's squeezing, to even things like glaucoma. You have these cells in the eye called trabecular meshwork cells that squeeze and relax in order to control the flow of fluid into and out of the eye. So yeah, huge numbers of areas where disease is related to force generation, but there was no way to screen for drugs to do this in high throughput, right? You have these academic tools, one-offs, one cell here, a few cells there, and then you have these diseases that are connected to force, but there's nothing to discover new drugs for those diseases. And so that's kind of where we came in and developed new technology, where we're measuring at scale, 96 well plate, 384 well plate levels, how much each cell is squeezing, and quantifying that very accurately for each of those single cells, and hundreds of cells per condition over plates that have maybe 10,000 cells or so.
So you've actually built two companies in this space, Avail, which you mentioned briefly, and Foresight. Could you tell us a little more about what both these companies are doing?
Yeah, so Avail has been working on this tool to measure the mechanical properties of cells. In the lab, we used to call this deformability cytometry. And so they've been commercializing that and are using it to measure the activation state of immune cells in patients that have sepsis. So it turns out that if you get a bloodstream infection or other infection where your immune system kind of goes out of control, it will start attacking your own body, and this is called sepsis, and this is a huge mortality and a huge burden to hospitals. A lot of patients that are dying in hospitals are dying because of sepsis. And actually, in COVID patients, they have a form, the patients that end up not doing well are usually the patients that have a form of viral sepsis, where their immune system overreacts to the virus and starts attacking itself. And so what Avail has been doing is a really rapid test using, squeezing cells, measuring thousands of leukocytes from blood, and being able to then say this patient has an activated immune system, is likely has sepsis, and giving a green, yellow, red. This patient has trouble. Be careful of this patient, or this patient is fine. And then doing that within less than 10 minutes, right? So very rapidly in a triage emergency setting, so that the clinicians or medical care providers can appropriately know what types of resources are needed for that patient. So that's the focus of Avail. They're in the middle of a trial now for that they will submit the data for 510 clearance of their system.
Awesome. Can you tell us a little bit about Foresight as well?
Right. Yeah. So Foresight has been commercializing the technology to do drug discovery based on cell force, or how much cells contract. And so they are they basically are their own drug discovery engine, right? So they are developing their own compounds based on screening a library that they have access to, about 200,000 compounds. And so they're kind of moving down in a few of these. The challenge with that space is there's so many diseases that have force involved in them. So which ones do you focus on first? So fibrosis is one area that there's a lot of interest in, just not great drugs out there right now. And there's going to be more and more pulmonary fibrosis in particular, given COVID. So they've been screening in that space and developing some nice compounds that seem to have effect on force generation and other follow-up assays. And so they're now continuing partnering with folks and kind of continuing that effort. But yeah, they have this beautiful platform that allows access to this new class of phenotypes that cause disease. So it's very exciting to see where that goes.
That is exciting. That's very cool stuff. So moving on to the last area that your lab is working on, quantum diagnostics. And so your lab is kind of pulling together some of the topics that we've mentioned to develop platforms that fully leverage our advances in our ability to analyze single cells and single molecules. Could you tell us kind of more about this platform that you're developing?
Yeah. So I think that the general framework on how we think about this, or what we call it, is kind of a lab on a particle platform. And so you probably maybe have heard of, the audience has probably heard of lab on a chip, right? This idea that you can, all the things that you would do in a lab, you shrink it down to, the size of a small chip, and fluids and moving around and washing, et cetera. So, I've been in the microfluidics field, and that's been one of the key concepts of that field. And to some extent, it's been great. It's enabled diagnostics that are been really helpful during the pandemic, for example. But in other aspects, there's limitations. And so one of the limitations that I've seen over my time, and even in commercializing these systems, is that for each new chip that you have, you need an instrument to run that chip. It's not like the chip plugs into what's already out there in your research lab or diagnostic lab. And even if there is a benefit from using those chips, for example, to create droplets and measure single molecules or measure single cells, getting to your point of the quantum diagnostics, there's still a barrier for people to use it. And the barrier is that you have to acquire new instrumentation, right? So that's one big barrier. And so that, that really lays it creates there's so many new things that you might want to test, let's say in the clinic, like what really low levels of biomarkers are associated with disease. But if there's not access widespread for these technologies, then you can't do these types of studies. And so So the approach that we took is what we call lab on a particle, as I mentioned, is using particles as a format to create small volumes, because all these quantum assays, all these quantum diagnostics, where you're measuring a single molecule or a single cell, it's all about compartmentalizing, creating a small partition of volume that you can put that cell in or putting that molecule in, so that when you do a reaction or measure something, everything accumulates to a high concentration at a small volume, and it's very easily detectable. So that's like the crux of all of these assays. And so our approach is using a particle to enable that. And so you can imagine, actually this is one of the particles that we use, or it's a 3D printed version of one of the particles that we use that, obviously it's like thousands of times smaller. In reality, it's about the size of a cell or so. You can see it's like a little bowl. So a little bowl that can hold a cell. You can put oil around it as well. And then you have now this small volume of fluid where the cell, let's say cell is secreting something, you can capture that at very high concentrations, thousands of times higher concentrations in that small volume of fluid. And you can now make measurements that you couldn't. And the key thing is these particles are compatible with instruments for analysis that are already in your labs. So things like FACS, flow cytometers, things like single cell sequencing systems that are already in your lab. And so that now eliminates that barrier to adopt these new technologies, and they can spread much more rapidly, because there's tens of thousands of these place instruments already that are able to analyze them. So that's the general concept there is using new technology, these lab particle-based technologies, in order to get to those fundamental limits again of biology, the single cell and the single molecule.
Interesting. And so you've talked a little bit about how these particle-based technologies can enhance equipment that we're already using, but how does that translate to functional applications? What are applications that these particles would allow that we can't do right now?
Yeah, no, so a good example is you want to often look at single B cells or antibody secreting cells. I'll give an example that's related to COVID because I know everyone can relate to what's going on now. So early on in the pandemic, we wanted to find monoclonal antibodies, and we still want to find monoclonal antibodies for variants that neutralize the virus, right? So instead of using convalescent serum to treat other patients, if you can create monoclonal antibodies recombinantly, you can use those as a therapeutic. So how do you do that? So one way to do it is you find B cells or other antibody secreting cells from patients' blood, or you immunize mice and make them produce B cells that secrete antibodies. And then you screen for antibodies that bind parts of the coronavirus, right? Spike protein. So how do you do those screening steps? Usually you need to do some type of secretion assay, or you make hybridomas so that you immortalize B cells so that they can secrete and you can measure the antibodies that they're secreting. So this type of lab on a particle platform allows you to capture the secreted antibodies from a single cell onto the particle, right? Captured onto the surface of the particle, and then add your antigen, could be the spike protein, let's say. And now if the spike protein binds and has a fluorescent tag on it, now you can sort out that B cell that has antibodies, that secreted antibodies specific to that protein. And then you can sequence it, right? And then you can identify what's the sequence for the antibody, and then you can feed that back into making the recombinant antibody. If it looks like it was a good sequence, then you can produce that therapeutically. And now imagine you can distribute these across the world, not just to a few labs that have much more expensive pieces of equipment that can do this, and be able to kind of democratize that discovery effort. So that's kind of one of the areas that we think it can have a big impact.
Very cool. And so you've also founded a company in this space as well. Could you tell us a little bit about what they're doing and how you think their mission of expanding access to you and improving single cell analysis is going to change the way we're doing biology?
Yeah. So kind of hinted at it in my answer previously, but the company is called Partilian Biosciences. And so they, in the end, they are selling these particles, right? These we call them nanovials because they hold nanoliter scale volumes, and they, so a single cell can fit into this. You can analyze functional aspects of that cell, whether it's secreting antibodies, secreting other cytokines, or interacting with other cells. So that's really the key there, is that this is, a vial of these nanovials, millions of these nanovials can be easily shipped and distributed across the world, across the United States, across the world. We've already done that, and easily applied by labs, whether it's pharmaceutical labs, whether it's academic labs or institutes that already have these equipment that flow cytometers or FACS instruments. And so they, from today, they can make advantage of this. And Partilian that's kind of their business, is really to give this and distribute this as widely as possible, because there's already this installed base of instruments that can use these reagents to really amplify their single cell analysis workflows.
That's amazing. Awesome. Well, we are coming close to the end of our time here, but a couple last questions before we close. First of all, where can people learn more about your work?
Yeah. So, I'm active on LinkedIn, so I'll post new things, like the latest things that are happening. That's probably the best place to look it up. My name is relatively unusual, so just Googling my name, you'll find all this information and then I have my academic website. It's not updated as often as kind of the latest things will probably be on LinkedIn, but I think those two places are a good starting point if you want to learn more about the things that we're up to.
Awesome. Before we sign off, do you have any shameless plugs or other closing thoughts? The one thing I want to say is that a shout out to all the bioengineers out there for all the great, if we think about the heroes of this pandemic, I would say the bioengineers are kind of at the, certainly we have our nurses and doctors and healthcare providers and the folks delivering shipments, but the bioengineers are kind of at the top of the list. You can amazing how rapidly new vaccines, new diagnostics were out there, and who's in response. In a lot of the cases, it's the bioengineers that are kind of the key people that are doing that. So, thumbs up to the bioengineers. Great career. All the young folks out there, think about that as one of your career opportunities. You can be a hero as well. That's very true. Awesome. Well, thank you so much, for an absolutely incredible episode. This was a lot of fun, and we're really grateful for your time.
Thanks so much, Cage. Take care, everyone.